You're very welcome today to this webinar from BioPhorum's ATMP Forum. My name's Kathleen O'Hagan. I'm a global change facilitator within the ATMP Forum, and I'll be hosting the webinar today. I won't be discussing the technical details, you'll be glad to know, but I'll be running the webinar today. And the webinar is entitled Leveraging Platform Process Characterization Data to Accelerate CGT Validation and Commercialization. And we have our three presenters here as well. We've got Jean Donovan from Regeneron, Benson from Sangamo, and Marcos from Bayer. So you're all very welcome, guys. Okay. The purpose of this webinar is to showcase a recent publication from the ATMP Forum, and the output that this team has produced will be discussed and showcased in this webinar. So in terms of the agenda, we'll start by introducing the team that produced the work. We'll give a bit of a background on why the team formed and the goals of the team. We'll then move on to an overview of the publication. So we'll talk about the regulatory landscape within this area and business considerations when it comes to platforming. We'll next move on to what is a platform, and we'll give a proposed definition that the team have suggested around what is a process platform and examples. And just to flag as well, so the scope of this work was very much around process platforming. We did decide as a team that analytical platforming was outside the scope of this work. So today's presentation will be focused principally on process platforming examples. Next, we'll move on to cell therapy considerations, and we acknowledge that there's a unique challenges when it comes to cell therapy platforming approaches, and we'll look at potential opportunities within this space. We'll then move on to phase-appropriate process development and characterization, and we'll talk about the sort of phase-appropriate considerations, and then we'll move on to the control strategy. Then we will close the presentation, time permitting, by allowing the three presenters to share their thoughts on what the future looks like when it comes to this area and potential next steps. There will, of course, be an opportunity to ask questions to the presenters and share your thoughts at the end as well. I would ask, if you are going to ask questions to the presenters, that you drop the questions into the chat, and we'll pick up those questions at the end of the presentation. Also, if you see a question in the chat that you're interested in and that resonates with you, if you want to give a thumbs up to that question, that will help us sort of prioritize sort of the questions that are most pressing and suppose need to be answered first. Scott, you have your hand up. Let me just unmute you. Hi, Scott. Are you unmuted?
Yes. Hello. I was just noticing that this app that we're running on Teams has both a chat and a Q&A function.
Yeah, I think
so.
I've noticed that too, and it's new to me, so it must be a new version of Teams. So maybe just into the chat, if that's okay.
You.
Yeah.
Thank you.
Yeah. I will, if people, I will check the Q&A as well, but let me just, okay. Right. Before we get started, quick disclaimer to say that the views and opinions expressed by the speakers and the team are their own and should not be attributed to the individual's employer, organisation, or other individuals. And very quickly, before we get started into the crux of this presentation, I do want to give a very, very quick overview on BioPhorum and the ATMP Forum, just for any members of the audience that aren't familiar with us. So BioPhorum is a global collaboration that brings together experts in the field to discuss common challenges and problems with the view of creating best practices and procedures that will help solve those problems and accelerate progress. The ATMP Forum is one of 12 forums within BioPhorum or specific discussion groups. You'll see us sitting here at the top. And we differ slightly from the other forums in that the other forums are focused on general biologics, where the ATMP Forum is specifically for ATMPs. So we discuss development straight through to commercialization within the ATMP space. Just to flag, some of you may have known us as the CGT Forum. We've recently rebranded to ATMP. If you notice in the publication that we're presenting today, we do refer to the CGT Forum, but just say we're the same forum, just rebranded, so just to flag that. And the ATMP forum, just to give a bit of background on it, it was formed in 2018. We currently consist of 31 member companies, and we operate at sort of two different levels of teams. So the first level is what we call these high-level discussion teams. And these are teams, quite large teams that meet every 2 months, and we really meet to discuss high-level topics and quick-fire questions. And we can be modality focused or functional focused. And then the second level of teams are what we call deep dive teams. So these are teams that meet more regularly and they are working towards a sort of tangible output or goal, or working on a specific problem. And the work that you're hearing from today is a result of one of these deep dive work streams. And I'll refer to this team just for ease as the Lev PC team, because I appreciate it's a long title. So why did this team form in the first place? This was really as a result of the increasing number of CGT products progressing through pivotal trials and towards commercialization. And this exponential growth in these therapies created a good opportunity and really, I'd say, a strong desire to look and understand common manufacturing approaches across these novel therapeutics, which in some cases may only differ by the genetic payload. So the team got together to sort of understand these common practices. And first of all, they looked at comparing practices when it comes to platforming in the general biologic space and understanding what we could apply to the CGT space. And then we discussed also additional sort of novel CGT considerations. And with that, then we summarized the different opportunities and published a paper to sort of summarize our discussions. This is some lovely photographs of our team members, some of the key contributors. The team consisted of 16 member companies, and we met every 2 weeks to sort of pool knowledge and discuss recommendations when it comes to improving efficiency for CGT product development. And we'll talk about some of those key elements in the upcoming slides. And this is the output from that team. So this is a publication that's available on the BioPhorum website. And I believe one of my colleagues is going to drop the link to that publication into the chat. So for those of you who haven't read the publication yet and are interested in this topic and interested in what you hear today, I'd highly recommend that you download and have a look at that publication. Here are some of the companies that were included in the publication and some of the authors and contributors to this work. And we have 3 of these team members on the line today. So we've got Benson from Sangamo, Gina from Regeneron, and Marcos from Bayer. And I do also want to acknowledge Kayla from BMS, who also did a strong contribution to this webinar, who unfortunately couldn't be here today. So I'm going to give a big shout out to Marcos, who was drafted in very last minute to support the cell therapy section of the discussion. So thanks, Marcos, for jumping in. Okay, we do have some questions before we get started into the details of the discussion, just to understand the makeup of the audience members. So I'm going to launch 2 questions, 2 live polls that will appear on your screen shortly. The first one is around what modality people from the audience are working on. There is an option to click multiple modalities, so please feel free to, let me just pull up this poll, to click which ones you are interested in, and I shall launch those now. And the results will appear on your screen. Sorry, the poll will appear on your screen and the results will appear in your chat. So we'll pick that up in a minute. And then the second question is really, we're interested in understanding what phase of development you're working on. So the options are development, preclinical, mid-phase, late phase pivotal, commercial, and other. And again, we'll launch those just to see the breakdown. And if you guys are able to click on the chat, you should see the results. I can see them creeping up now. Okay, so good number from cell therapy, Marco. So 20 from cell therapy, 25 from ex vivo gene, in vivo gene therapies 32, general biologics 16, and other. So I'd say pretty much an even spread across modalities, so that's good. And then in terms of phase, again, mostly early phase, I'd say. So we've got 19% development, 22 preclinical, 24 mid phase, 18 late, and then 11% commercial. So thanks for that, guys. That'll give us a sort of a good indication of the makeup of the audience. Okay, you'll be glad to know that that's me for a while. So I'm going to pass over to one of the key authors, Gina from Regeneron, who's going to start talking about the regulatory considerations.
Thanks, Kathleen. I will start by noting that I am not the expert on this regulatory section. The main contributor for this portion of the paper has left the team, so I'm stepping in and trying to do this section a little bit of justice as a stand-in for him, but just keep that in mind that I am not the main contributor on this topic. So regardless of therapy type, there are regulatory requirements that must be met in order to successfully commercialize a new product. The regulators expect your process to be defined. So, what unit operations you have, the order, and what the purpose of those is, characterized so that you really understand how and why your process is running the way it is, and validated to make sure that your manufacturing process can reliably produce a quality product and ultimately ensure patient safety. Next slide. So in tandem with regulatory expectations, there are some business risk decisions that can be considered when developing and commercializing a manufacturing process. Cell and gene therapy products are still relatively new in the industry, and therefore, some of the biggest hurdles can include these examples outlined here, such as limited product knowledge, decreased material volumes for development and characterization studies, and a limited number of manufacturing runs. So as a result, the product and process understanding can be a challenge to really fully define prior to a commercialization. And the clinical readouts and data from these products can be so incredible that the speed portion of this may be more expedited than we would expect from a traditional biologic. Next slide. So I'm not going to go through this, but this is just a summary of the ICH guidances that we were heavily consulting for this paper. So we looked at Q8 through Q12. These were significant reference documents while we were going through and authoring this paper. There's also a lot of additional regulatory guidances from FDA and EMA that are specific to cell and gene therapy that are also included as reference. It's important to note here that ultimately we feel it is the responsibility of the sponsor to engage with health authorities prior to commercialization to ensure that any unique or creative packages that are going to be submitted are going to be accepted at an alignment with regulatory expectations, and really coming prepared with specific information and questions to those meetings will provide the greatest opportunity for actionable feedback from regulatory bodies. So coming back to what we mean when we say business considerations, there are some studies that may not be required by regulatory bodies at a particular stage in a product lifecycle, but performing them earlier on versus just performing them prior to commercialization can still mitigate risk to the business. So studies that might fall into these buckets should be assessed for applicability to a particular program and what level of risk is really acceptable to the sponsor. When assessing that business risk, we wanted to highlight some of the CGT specific challenges and realities that companies are facing today. So these can include, a product portfolio that's still primarily in early clinical phases, financial realities such as consolidations and acquisitions, the role that CDMOs play in this space, and really a highly competitive intellectual property space that can kind of limit some knowledge sharing and collaboration across the industry. So moving into platform discussions. One of the ways in which new therapy types can take and modify learnings from other biologics like mAbs is with respect to process and product platforms. In the monoclonal antibody space, process platforms have been shown to be highly effective. And the team performed a survey to try to better define what is a platform. And we are showing the results of that here. For the context of this paper, we're referring to an and/or here, right, where we have a production system, including process conditions, raw materials, and technologies, and/or a related group of products that might share chemical and physical properties. The ultimate goal of having a platform is to be able to take any number of pieces of a process or a product and make them essentially a starting template, which can pave the way for similar programs in the future in the hopes of accelerating development and characterization. Now, this doesn't mean that the future products don't need the same level of characterization and understanding, but rather by understanding those templated pieces ahead of time, you're able to set yourself up to prioritize and focus resources on developing and optimizing those areas or attributes that are known to be different between products. So this may be something like changing of a transgene in an AAV process or changing a cell collection source in a cell therapy. The team does not acknowledge that leveraging and platforming in cell therapy is more challenging just due to the nature of the processes and products. So there is a heavier emphasis on opportunities and examples for gene therapy that are discussed in the paper. So again, continuing with the gene therapy examples, we put together a high-level process flow and looked at what might be applicable and like what approaches might we be able to apply from other biologics like mAbs. And we outlined those in orange. So you'll see, things like cell lysis, affinity chromatography, cell thaw, and impurity clearance. Those are things that we can take our learnings from mAbs and really apply them to the gene therapy space and hopefully get a robust platform going. The steps that are outlined in red are more specific to gene therapies. So these are, things like ion exchange chromatography, plasmid. Obviously, we don't have plasmid for mAbs, and nanofiltration. There's a bunch of other different considerations that could still be platformable, but will be more specific to the gene therapy space.
So we are going to pause here to hear your guys' thoughts on unit operation that you are considering when it comes to platforming approaches. You'll see we have a mixture of sort of gene therapy specific and ones that can be applied also to mAbs. Again, I'm going to launch another poll and it'd be good to hear your thoughts. So it should be coming onto your screen now. So the options we've listed are platform production, cell banking, transfection processes, cell lysis, affinity chromatography, ion exchange chromatography, nanofiltration, mixing studies, impurity clearance or others. If it's other, it'd be good if you want to drop into chat the other options. It'd be good to see that. And we'll pick up the responses for this maybe at the end of Gina's section. So I'll pass on, Gina, to you.
So just again, continuing with some of these gene therapy examples, the paper outlines more specific areas that might be open to platform processes. So these might include strategy for viral safety testing, expression system, media development, seed expansion of a host cell line. So, prior to transfection, if you're using hex cells, you can, that seed expansion process can be the same between multiple products. Plasmid production, again, similar idea where you're just changing the plasmid gene and the fermentation may be platform product to product. Chromatography and general process considerations like raw materials and equipment, as well as sequence of operations. I think when we talk about platform, sometimes the manufacturing side of the platform can get kind of overlooked or lost. And it is still really important that, the end commercial process is kept in mind when developing these products, right? If you have a completely different mixer between two different programs, you're not going to be able to have like a platform mixing validation, for example. So continuing with a more specific example with transfection, we considered this one in a little bit more detail again in the paper. And the idea here is that you can turn as many parameters as you can into constants and then focus heavily on those known variables. You'd still have to do a check the box to make sure that those constants are working for a given product. And if they don't, then that would require additional work, but it would at least be a little bit, ideally, I'd be less resources that you would need to sink into those parameters. So in this case, we're talking about transfection, and the platform process may include the same RepCap and helper plasmids, complexation media, raw materials, such as bioreactor type and size, transfection reagent, and expression system between all or a portion of products within a given portfolio. And then your main variable really becomes that transgene, and you can focus a lot of the development efforts on parameters that might exhibit gene of interest specific sensitivities. So some examples that we thought, were probably value added to consider when talking about those gene of interest specific sensitivities are things like, cell density at transfection, plasmid ratio, complexation time, and among others. A lot of different parameters might be affected by the gene of interest specific plasmid. So it's really just being able to come into development with an idea of what parameters might be impacted, and then, targeting the development work from there. And parameters might be, even within these examples, if you're not seeing a difference with different gene of interest, you might consider that platform after multiple sets of product data and demonstrate that you have consistent product output. And generic examples to consider for platform capabilities can be taken from other biologics where we have an established knowledge base, which can really greatly reduce the number of variables and promote future success through commercialization. So this is just a nod to some of these potential learnings that could be carried through into the CGT programs. And with that, I will hand it over to Marcos to go through some of the cell therapy considerations.
Thank you, Gina, and once again, thank you for the opportunity to participate in the webinar. And when we pause and we reflect over what can be the biggest challenges for the cell therapy in terms of platform development, I will in the way rephrase what Kathleen just mentioned in the beginning. Very unique. So we are looking for basically four main pillars around it, these specific challenges, and we can go from the heterogeneity of the cells that we can have, since we always rely on the initial donor. We know that specifically in terms of this approach, we can lead into a very limiting product quantity and stability, which allow us to understand how we can go from autologous approach down to allogeneic and more, I will put in between commas, universality approach. And this definitely will lead to one very important part, which is the fact that we need to mass produce, and we need to understand that now the product is the cell, and the cell is not producing our therapeutical products, such as, for instance, a gene therapy viruses. And in here, we need really to be paying special attention and efforts, because this product is sensible to the processing conditions that you have in the platform. The last pillar that we have is the one that is mentioned on the down left side, which is precisely the technology that we can use to run the processing of the same cells. At this stage, we know perfectly are aware that there is much ahead of us that we need to learn, and also that we still are in the early stage of the experience, and there are some limiting information in here. Well, basically what the approach is many times is that we do not have a tailor-made technologies. We have in fact a technology that we adapt from other processes that we have been learning from and using on this space. But when to the next slide, please, Kathleen. But when we're looking into very specific examples, you can always thinking about how modular can be our processes and how far can we leverage that same modularity to apply in different circumstances. In these slides, what you can see is three major examples. So we're looking for the autologous approach, in which we go from patient to the same patient. We're looking for the possibility to also have a healthy donor. And from here, we can try understanding and develop a allogeneic approach. And in fact, and again, between commas, the more universal possibility, which is the third example, the one on the bottom for the iPSC allogeneic approach. One thing that is really, really interesting to see in here is the common factors. So allogeneic, autologous, and healthy donors, you can see that the phases of cell sorting, activation, and gene transduction and gene editing are common to them. So we can learn with it. We can apply one from to the other. When you're thinking about then going for the iPSC allogeneic, in fact, what you have here is more on the scale itself, on the expansion and differentiation phases. And this is where we can learn from this iPSC allogeneic and how we can bring in the modularity way some of the different steps that we have in the different approach to learn with it. One last comment, and very, very important here, and also rephrasing the beginning when Gina in this webinar, and Gina mentioning the importance of quality. So when you look into the right side, you can see that it all ends up to quality. Quality assessment is critical because cells needs to be in a very perfect state to be used for the therapeutical purposes and not be a lead cause for any type of diseases that they cause from degeneration of the cells itself. So I think that in terms of the modularity approach, and if we're thinking about iPSC's allogeneic, and I will ask for the next slide, please, Kathleen. When you're looking for the opportunities that we really have in these more allogeneic therapeutical and mostly based on the use of pluripotent stem cells, iPSCs, we can see that there are four major stages when we can learn from it. On the beginning, clearly there is the need to mass produce cells, and this is the part of the stem cell expansion phase. And in here, there are many similarities in between the different cell types, the different iPSCs that can be leveraged as a unique platform to run the cell expansion. When you're thinking about, and I will jump to the third box in here, when you think that after expansion, there is the need for differentiation, then it comes different type of requirements. Scott also mentioned here that in terms of platforms, there is in the chat, you mentioned the chat, the 2D and 3D. So when you're looking into different differentiation products, we might have the need to leverage this type of platforms. If you're looking for a neuron, we're going to need to have a 2D approach. If you're looking for a cardiac cell, we might already have a 3D approach. Anyhow, we can always learn from that. Independently of the platform for the expansion and differentiation, one thing is for sure. When you look into the fourth box, the last one, and for the downstream processing, definitely this requires fast times and fast processing without influencing the quality. Because in here we are dealing with cells, and cells will be out of their own environment. Cells will not be under specific conditions controlling oxygen, pH, or even have access to nutrients. So cells will not be in their natural environment, and with that, we need to go fast. The second box was not the second box. Sorry, was not missed, or it was just postponed on purpose for one reason. When you're thinking about mass cell expansion, or you're thinking about our final product, one thing is for sure: we need to have a filling process. We need to have the product formulated for therapeutical purposes. And in here, we know that there are different specific products as well, and there are some maturity and the need also to revamp some current technologies that are being used for final fill and finish and storage prior to applying to the patients, that we can learn from it. So at the end, in these three slides, basically the idea was to go from a more broad perspective on the cell therapy, in which we could include in the same pocket, sorry about the expression pocket, but in the same side both autologous and allogeneic approach. Then we could understand where these different two approaches can have common steps, and we end up in this more universal-like approach, the iPSCs, in which we can really understand how to have a strong platform development opportunities. And I think this is it. Kathleen, I'll pass to Benson. Thank you.
Awesome. Thank you, Marcos. And I think that's a it's a great transition to what I'll be talking about, especially coming from cell therapy and gene therapy company where we're trying to do both, and we can see the differences in terms of when you're working with gene therapy versus working with cell therapy. So thank you, Catherine, for getting to this slide. So the next part of the slides, as far as the phase appropriate studies go, I will mainly focus on gene therapy, and that was actually by purpose, on purpose, mainly because as you heard from Marcos as well as from Catherine from the beginning, it's fairly hard to do the same thing for cell therapy. But for gene therapy, there is a few things that we can still be able to leverage from traditional biologics. And in addition to that, I do feel like a number of companies they have done way more on the gene therapy compared to cell therapy. And I think having this kind of webinar, I think it's also a good starting point for people who are actually working on cell therapy to come together and hopefully start putting some similar information for gene therapy. So I think it's a good transition for that. Okay, so the next few slides I'll cover some of the things that we talked as a team to see, well, we know that there are some things that we need to do overall during the development of the molecule. We also know that, as Gina mentioned, we have many opportunities to platform processes, and also, again, in conjunction with that, platform a number of other things that Catherine started talking about at the beginning, including things like assays. But the idea of doing that is to really be able to see whether we can be able to use all this information to get to the validation and commercialization of the molecule faster, especially for cell and gene therapy. So the next step that we took is, as a team, we came together and from different companies, what are the and started thinking about what are some of the activities that various companies do to assess or to make sure that we are ready during the process development as well as commercialization of CGT. And then based on that, try to see, okay, we know that there is regulatory consideration and also there is a business consideration. How can we be able to use those opportunities to accelerate a molecule development to get to the validation and commercialization sooner? So a few things that as a team we were thinking about. The first one is we want to, in general, and I know this one can get a little tricky, we want to have the final process or the process designed to make the molecule that we are making as soon as possible. Well, we know that sometimes there will be challenges because, again, trying to balance between the front loading of activities that we need to do. But it's really important to do that. And it's important to do that for two reasons. The first one is the historical data, and as you're building knowledge, it makes it easier for you to know what you actually, how your molecule and how your process look like. And we'll get to that on the control strategy part. But the other part that is also important is how do we mitigate any changes that we make along the way. And if you have a process that is similar, let's say during the clinical process development, early stages and late stages, and you go with that to commercialization, it actually is a much smoother transition when it comes to comparability of that particular molecule. So that's something that we did consider in this. I did talk about the weighing that the cost and resources of front loading those items, and therefore, and this actually can be taken as a from one company to another company. Some companies are okay doing that. Some companies might not be too interested in doing that, depending on a number of factors that Gina raised when we were talking about platforms. And then based on the brainstorming session, we look at the upstream, which is shown on the first column. And we also looked at the downstream activities as well as the fill finish activities that various companies do for the reasons that I mentioned previously. And then so that we can have an idea of, hey, this is where we're starting. how do we bucket things so that we know what we can do earlier and what we know, what we think we can platform, and also see whether there are opportunities to be able to, again, from as we are going through the development process, where we can put what. So just to kind of, again, this is just a list of a few items. So for upstream, as an example, cell line qualification is one of the most important part to do. So I'm highlighting that. The other one, and I think it was raised during in one of the questions, the poll question, is the GMP plasmid sourcing and how the robustness runs of that one actually look like. The other one is process-readed extractable leachables. And I'm putting this one because I'll come back to it in one of the later slides. And again, the list that we're looking at here is in the paper. There is many more in the paper, the link that was provided. We're just highlighting a few. And there is many more, maybe, or even some that could be less depending on the molecule. And then the middle column is why we have the downstream process. And for the downstream process, of course, we do have to think about things like mixing studies, because if you have let's say a unit operation that requires you to have homogeneous material, it's very important that you have that. And Gina also highlighted this when she was talking about platforming. Another one, especially for downstream processes, is for CGT is impurity. So if you have anything that is unpackaged, so we've been talking about empty, full, and all the other variants that you can see with the AAVs, it's really important that you put that into consideration as well. And then back to my favorite topic, extractable and leachables. And the reason why this is important, as you can see, it's popping again in downstream, and especially since when you're working with CGTs, and I'll get into this in just a minute, a lot of materials that we used are susceptible to extractable and leachables because they are polymeric. And then finally, we have the fill finish. And the fill finish, this is, you're getting closer to the patient, and therefore the opportunities that you can be able to see from the very beginning, and you have, and you can take those opportunities to control them. You really reduce the risk of having molecule that is either, the product quality is impacted, or it's not efficacious to the anymore. So the fill and finish also becomes quite important. All right, so Catherine, if you don't mind, can you move to the next slide? So as I mentioned in the previous slide, after we looked at all the, well, not all, but most of the items that different companies do, and we have a list of what is being done during the upstream process, and we also have a good understanding of what is happening in downstream, as well as fill and finish, we go to the next step. And the next step is, as we had previously, we as the drug developers, as the sponsors, we are responsible to make sure that the molecule that we're making is safe and efficacious for patients. And that's what the regulatory agency really is confirming, to make sure that we're doing that. And it's really, again, it's our responsibility to do that. So that is the goal that we have in mind. The other thing is that the regulatory agency is aware that as we're going through the development process, at the very beginning, when you're doing our tox studies and phase 1 studies, the requirements, we're still trying to understand the molecule. We're still trying to understand the process. And therefore, there are some things that we can be able to, let's say, leverage or use risk assessment on. And on the other hand, as a company, you may not be willing to spend a lot of time at the very beginning of the molecule because the funds can be challenging. But again, the bottom line is that we still want to make sure, and it's our responsibility, that we're producing safe and efficacious material. And then based on that, we looked at, well, again, from the previous slide, we have all these studies that we need to do. From the requirements that we have from the regulatory agency, can we be able to either develop some things earlier and also leverage that to other molecules, or can we even wait after commercialization, depending on let's say the classification of the molecule that you're working with? Because in some cases, if you're working with rare diseases, you may be able to do some things later on. Okay, and what we're showing in the table below, again, kind of similar concept. We have the first column. This one is talking about phase 1 or 2 enabling studies. And if we look at the upstream as an example, if we really want to understand, again, from a regulatory point of view, cell line qualification falls under that bucket. And then same thing applies to, let's say, impurity clearance for downstream, as well as, let's say, filter integrity test, where the technical knowledge and the regulatory guidelines really emphasize that we need to understand those three things even in phase 1, phase 2. While as for other things like mixing homogeneity studies, that becomes more of a business risk, and therefore for phase 1, phase 2, maybe it might not be so much of an emphasis, but if you have any platform work that you can be able to leverage, it really reduces the overall business risk, and therefore, if you have the opportunity to be able to do that, you go ahead and do that. And also another thing that I wanted to emphasize here is that with the CROs or CMOs, if they have a way of platforming the overall, let's say, equipment and the production process, this is another one can be quite important when we're moving to a CMO. And then as we progress to phase 2, phase 3, again, at this point, you need to have a better understanding of your molecule and your process than phase 1, 2. We do something very similar. So for upstream, we have some studies that we strongly feel from regulatory guidelines, as well as the technical knowledge that needs to be understood at that point. There are other things that are included there. And same thing with the business process. And then at the very end, so to the right side of the farthest column, this is where now we're getting to launch enabling. This one gets interesting, and the reason for that is at this point, you really need to know how you need to have a very good understanding of your process as well as your molecule, because at that point, you're looking for consistency, not just from your product quality point of view, but also from process. And this becomes a part of supporting the qualification as well as control strategy. So Catherine, if you don't mind moving to the next one. Okay, so I didn't mention about extractable and leachables today because this is one of my favorite items. And the reason for that is, again, for cell and gene therapy, we have been trying to move to disposable systems. They're flexible. You can have them in different sizes. You can do a quick turnaround of your from one production batches to another production batch, but it does come with some limitations or some challenges, and one of them is extractable leachable. So an extractable leachable, the other reason why it's really important is because you have to put a lot of stakeholders, and you have to look at it from development from the very beginning, from the time that you have with in the upstream process, all the way to fill finish. And the risk is different. So really, what you're looking at here is a table of when we are performing our risk assessment, that guides us to inform us whether we need to do a study or not. We can look at just the overall, let's say, the steps of production. So we have drug substance, we're making the drug substance, we're storing the drug substance, we're going ahead and filling the drug product, and so forth. But in addition to that, we have to look at extractable data, let's say that is coming from the vendor of the single-use system. And we look at the extractable data, and that will inform us of what we need to do. So from platforming point of view, you can be able to get, let's say, one molecule to another molecule, and as long as you can show those, there are either similarities in terms of how the solution behaves, you can use that as a platform approach. And in addition to that, for anything that is happening in upstream all the way to downstream where you get to the UFDF process, you can risk assess that based on the fact that when you go through the UFDF process, you have the opportunity to remove most of the leachables that you may have overall. But as you're getting to the drug product, things will change. So this is one of my favorite risk assessment topic in general. All right, go to the next slide, Catherine. Awesome. Well, when we started these efforts, we did talk about platforming processes and the following a phase appropriate development approach. And initially, we were actually even wondering, do we want to include control strategy as part of this effort? And we realized that when it comes to control strategy, especially for CGT, it's complicated. And it's complicated, as I'll get to the next slide, because of a number of factors. And those number of factors are driven by, if we start looking at what is control strategy. Control strategy is we're trying to understand our molecule. Again, going back to what I mentioned earlier, we're trying to understand our process. And based on that, determine, okay, when we get to commercialization, what kind of testing, as an example, that we need to do. How can we be able to show consistency even though we have had limited information overall? So it will bring all the other key stakeholders into this mix to make sure that our control strategy, once we get to commercialization, is robust. Okay, go to the next slide, Catherine, please. All right. So, and for CGT, I mentioned that is complicated, but we can reduce that complexity by overall using, again, platform data, as well as we are going through the development process, we are generating data that allows us to be all set for kind of putting our control strategy later on. That is despite the following. The first one is, yes, we do have a very small data set, and because in cell and gene therapy, we don't have a lot of, let's say, lots that we need to, we produce. The other thing is that even when we have limited material, the number of assays in general that we have to do is quite a bit. But it's still important for us to keep that in mind, that we're going to need that data later on when you're doing when you're setting the control strategy once we commercialize the molecule. The other thing is that the process complexity overall, and that actually is a great differentiator of gene therapy versus cell therapy. It can cause some challenges when we get to putting together the control strategy for commercialization, because again, all that information, it helps you, but if you're not careful, you're not going to have it when you get to commercialization. So it's really, really important to start thinking about it from the beginning. And then the other thing is that the assays also becomes quite important. And I think this is one of the reasons why at the beginning of the webinar, Catherine mentioned that assays is out of scope for this discussion, even though it's a very critical part of getting to know what you are going to include in the control strategy. So just something that we have to watch out. And that pretty much covers the control strategy. So I think the next one is Catherine.
Yeah, so I suppose that brings to the end of the presentation and the content of the publication. The team thought it'd be good to finish on this quote from Peter Marks from the FDA. And really, this quote talks about the idea that if you have identical vector backbone, that's the same across several products, and you characterize manufacturing of the first couple of products well, then in theory you could potentially say you have a platform which will allow you to focus in on just the critical issues for each product. And we are aware that there's an expectation the FDA will be releasing draft guidance around platform designation shortly. So I think it's something that's very much on their radar, and it's something that we need to watch this space. We did decide that we were going to end with the presenters giving their thoughts, but in the interest of time, what I wouldn't mind doing is just picking up some of the comments in the chat, if that's okay, guys. Alexis, you've an interesting comment question, so I'm just going to see if I can unmute you. Maybe you can speak to it. Alexis, are you able to come off mute and maybe talk to your comment? If not, I can read it out.
Hey, sorry, can you hear me?
I can, yes.
Excellent. So, so I'm a freelance regulatory consultant in the area of advanced therapies. And it's my experience that for a US marketing application, you have to provide a very large amount of source documentation, copies of source documentation with your BLA. And that includes all of your documentation related to your control strategy. Now, if you've supported, for example, proven acceptable ranges with another product, I would just add that you need to think about whether you can access that other product documentation, and obviously, therefore, whether you own it as well.
Yeah, I think that's a great point. And all of the, so the example that just immediately comes to mind is, seed expansion for hex cells, right? Like if we're saying that that's platform product independent, I would almost call that product agnostic. And the way that those reports are drafted should reflect that, right? Like we shouldn't be contributing it to a specific product if it can truly be a product agnostic study. But I completely agree. I think if you're using it to define PAR studies and it's based on like actual program specific data, I think that's a tricky spot to be in.
And yeah, and then to add to Gina's, the other thing that that definitely can help during the clinical development, you can reduce the business risk, Alexis. But I agree with you. As you're getting closer to process validation and commercialization, if you don't have that data that you use to support earlier stages, you definitely need to find a way to be able to put that data package together, because you're going to need it. You're going to need it for filing.
Any other thoughts there, Alexis, on that?
No, that's great. Thanks.
Kathleen, so apologize. I don't want to revert and be the one making questions, but the curiosity to Alexis to her comment. You mentioned that this package documentation is mostly asked on the US. Is the same situation also in Europe?
So the EU tends to work a little bit differently to the US. The US wants to get very much deep into your details, into your data, review it. The EU tends to be lighter on the provision of any type of source documentation, but obviously you have to put enough detail into your marketing application.
Thank you.
Does anyone else have any questions? Feel free to put up your hand and we can unmute you if you do. I don't know, is Scott still on the line? No, he had some comments. I've got Jason. Let me find you, Jason. Let's see if I can unmute you. There we go. If you should be able to speak now, if you unmute yourself. Are you able to speak, Jason?
He's on mute.
I've allowed your mic, so if you unmute yourself, you can speak. While we are waiting for Jason, just say we do have a few questions that came in prior to the call. We have 5 minutes. Let's see if we can get this one question done. So one of the questions was locking down at pivotal appears to be a real challenge. Do we know why and how can we alleviate these challenges? So I'll open up to the presenters. And I see another questions just come into the chat as well that maybe we might have time to address. Or Alexis, you've just got a further comment on that. So Gina, do you want to maybe, or Benson or Marcos, take that question on the screen, and then we'll just leave it at that.
Yeah, I mean, I think since a lot of these therapies are targeting rare diseases and, there's a limited patient population, there's limited material that you actually need to get into the clinic, which means fewer number of data points from manufacturing. So you're working with a small number of batches, and then combined with that really promising data from the clinic that we're all hoping to get, we can result in really moving very quickly to commercialization with increasingly smaller data sets. And I think that kind of poses that challenge for locking down your process, right? Like we're all trying to get drugs into clinic as quickly as we can, and then maybe that process isn't as optimized as it could be for commercial, and then you're trying to make those optimizations as you're speeding toward a commercial application. So I think platforming can help with some of that, right? Like it can kind of alleviate more of those, you know, if you can optimize a piece of the process that will work for multiple products, it can help you in the long run. And same thing, like really understanding what data you can leverage from your development package and what you need to support that application.
Great. Thank you very much. I just want to very quickly say that the discussion within the ATMP forum doesn't end there. We do have a members-only face-to-face event in June in Rockville in the US, and Benson, your two favorite subjects are on that, so extractables and leachables and platform approaches. And we do have a full day dedicated to platform approaches, both discussing analytical and process applications, which will be a mixture of case studies and working sessions. In addition, I mentioned the deep dive teams that we have ongoing with the forum, and we are investigating, all the deep dive teams are very much member-led, as the whole agenda is. We're discussing a possible follow-on team to this piece of work and understanding specifically what are the topics we want to discuss for further discussion. So watch this space, and if anyone is interested in continuing that discussion, please absolutely reach out to me. It'd be great to hear from you. We have 2 minutes left. I'm going to let people go. I just want to thank very much the presenters and our entire team. It's been